Welcome to the Organic Chemistry Podcast, Dr. Brian Lloyd's Scribblecast of Organic Chemistry lectures and solutions to homework problems. In this Scribblecast lecture, we are continuing our look at classes of lipids and looking at class number 5, the prostaglandins. Prostaglandins are a family of naturally occurring 20-carbon fatty acids that contain cyclopentane rings. So we have 20 carbons, a cyclopentane ring, and a carboxylic acid. And that's a fatty acid. These are synthesized in the prostate gland, lungs, liver, uterus, and other organs. They're found throughout the body and are believed to play a very important role in hormone regulation. They have extremely potent biological activity in fertility control, B, muscle contraction, C, blood pressure regulation, D, blood platelet aggregation, and E, suppression of gastric HCl production. Now, prostaglandins play roles in labor induction as indicated in 1 and 2. 3 and 4, or A and B. C and D, 3 and 4, have stimulated research in the use of these compounds in the treatment of heart attack and stroke. Prostaglandin is also thought to modulate renal blood flow, and as a result may have some role in kidney problem regulation. Aspirin interferes in the biosynthesis of prostaglandins. So it interferes or suppresses biosynthesis of prostaglandins. It is possible that the reduction in the rate of prostaglandin formation may result in a reduction of a fever and the relief of pain. However, a side effect of this, of course, is increased production of gastric HCl, which can be a potential problem for those suffering from ulcers. Because of these and other important biological functions, prostaglandins are often considered as one of the most important class of biologically active compounds ever discovered. Let's take a look at their structure. All prostaglandins, remember, are carboxylic acids that have a cyclopentane ring and are composed of a basic 20-carbon skeleton. Hence, they're based on the following structure, known as prostanoic acid. So we have 1, 2, 3, 4, 5, 6, 7, and I'll make this down, so I'll put this to a wedge, and we'll make this a cyclopentane ring. And that's carbon 13, 14, 15, 16, 17, 18, 19, 20. Okay, and the last one, of course, is a CH3. So if we were to number, we'd have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, and 20. This molecule is known as prostanoic. Acid. And as you can see, it is a carboxylic acid, has a cyclopentane ring, and 20 carbons. Now, prostaglandins can be broken down into several subclasses based on how they vary from the prostanoic acid derivative. For example, if I'm going to just use this side chain, the first side chain as R prime, and the second side chain I'm going to put a double bond here, and then we'll put our double prime here. This is an OH. If there is an alkene and a double bond O here, this is known as PGA sub 1. Now, let's identify what these designations mean. The classes of prostaglandins are grouped under the designations A, B, C, D, E, F. These all differ in the number and location of double bonds and the substituents of the cyclopentane ring. The PG, in this short form, means prostaglandin. A has this types of structural framework. The 1 means the number of double bonds. Okay, so let's continue. If I draw my cyclopentane ring again, I have the double bond O here. My R prime. Again, I have the same double bond here. R double prime. But you'll notice the double bond in the cyclopentane ring has changed position. It's now located here. This is PGB1. Well, as you might guess, I'll just draw it exactly same structure, 5-membered ring, R prime. Again, the exact same side chain, R double prime. Here's my OH. But this time, the double bond's located here. Well, that's PGC1. So, if you have a ketoalkene in the cyclopentane ring, a ketoalkene in the ring is either PGA, PGB, or PGC. The position of the double bond in the ring is what determines the A, B, or C. What's interesting is the variation in functional groups off the ring when you go to D, E, and F. If we get rid of the double bond and turn this double bond O into an OH, and have a keto alcohol that looks like this, you have PGD1. If, however, the keto alcohol is switched, and again, notice there's no double bond in the 5-membered ring. This is PGE1. And lastly, oh, it looks like for the PGC one, I forgot the keto. There should have been a double bond there for PGC1. So there's PGC1. Don't forget the keto alkene. In PGE1, there's no alkene in the ring, and there's no keto at all. But rather, you have a cis diol off the ring. Oops, sorry, that's PGF1. So, PGD and PGE are both keto alcohols off the ring. They just switch positions, and PGF is the diol. Now, sometimes there is an alpha beta designation after the number. And this can occur on rare occasions. Alpha means the OH on C9 is cis to the carboxyl side chain. Well, look up here for the numbering. C9 is this position. So PGE can't have an alpha designation. But if R prime, the carboxylic acid side chain, is down as indicated in the dash, and the OH is also down, it is alpha. So this one could be designated PGD1 alpha. Likewise, keto alcohols labeled PGF1 would also be alpha because the R prime and this OH at C9 are cis. Beta would mean they are trans. All right. We can practice some nomenclature. I'm going to draw a few of these prostaglandins, and I want you to use the PG designation to name them. For example, if I drew this one, what would this be? Well, diol off the ring is designated as PGF. There is one double bond. You'll note that double bonds in the ring aren't counted because double bonds in rings give you the PGA, PGB, or PGC designation. So they're already included there. It's only double bonds outside of the cyclopentane ring that are counted. There is one double bond, And the OH at carbon 9 assists with carboxyl, so this is PG1 alpha. Let's try another one. What would this one be? No, always confirm that there are 20 carbons in the structure for it to be a prostaglandin. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Clearly, this is a keto alcohol with a double bond O at C9. That is a PGE. It can't be alpha because the double bond O is not cis or trans to the carboxyl. Also, how many double bonds are there? There's 3 double bonds. Notice you only count double bonds outside of the cyclopentane ring. So this would be appropriately indicated as PGE3. Well, how are prostaglandins synthesized? If the prostaglandin is actually made from some 20-carbon fatty acid, such as eicosatetraenoic acid, perhaps there's unsaturated bonds there. How is it done? Let's take a look at the proposed mechanism. The carboxylic acid so we have a CO2H and we have a few double bonds in this chain. Now let's make sure we have 20 carbons. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. And a reaction takes place with oxygen gas. Now oxygen, we've learned, is a diradical. Because of this, one electron from the diradical can go here, and one electron from the diradical can go here. It grabs one electron from the pi bond, and what would happen is you'd end up with a radical in this position. Now, I can draw that just quickly. Boy, I'm having a bad time drawing straight lines. So, okay. So if that actually happened, you would link up here, get a peroxide, and have a diradical here. Now, radicals are not stable, but the orientation is optimal that in the presence of the enzyme cyclooxygenase, okay, so one word, oxygenase. So cyclooxygenase, not only is the peroxide link made, but these 2 electrons couple to close here. And when that happens, you actually get a cyclopentane ring with a peroxide in it. Also, the cyclooxygenase with the peroxide alters this double bond so that a peroxide as well. Now, if I was to redraw this as it appears in your notes, I now have my 5-membered ring with this peroxide linkage. And I have that dashed. Which looks like this. And then down, double bond here, down. Nine, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. And the peroxides here. So these two molecules are the same thing. And cyclooxygenase creates this peroxide unit. Okay. Now the peroxide unit is converted by an enzyme called peroxidase to a regular alcohol. And this group becomes an OH. Now, if you do endoperoxide isomerase, depending on the nature of the enzyme, whether it's reductive or oxidative, the peroxide will either form a keto alcohol or form a diol. If this bond breaks, and you get two OHs, you've got your PGF. And with these two down, that would be it. However, if it breaks and you get a double bond O, you get your keto alcohol, you produce a PGE. That transformation is caused by an enzyme called endoperoxide isomerase. All right. Prostaglandins, because of their importance, have the potential of becoming one of the most important class of biologically active compounds ever discovered. They're biosynthesized enzymatically from a C20 unsaturated fatty acid, such as this fatty acid at the top of our slide here. 5, 8, 11, 14, eicosatetraenoic acid. Remember, aspirin inhibits prostaglandin synthesis. It acetylates this cyclooxygenase enzyme and deactivates the enzyme. So, because of this, the actual synthesis of the prostaglandins is inhibited by aspirin, and the enzyme's deactivated. Well, so far, everything we've looked at has involved lipids that are derived from fatty acids. Even prostaglandins have the long-chain carboxylic acid unit. However, fatty acid-based lipids are not the only lipids. Because fatty acids can be deprotonated to make soaps, fatty acid-based lipids are called saponifiable lipids, meaning they can be used to make soap. There are lipids that are not based on fatty acids, and because of such, they can't be used to make soaps. They're called non-saponifiable lipids. This whole different class of lipids is what we're going to take a look at in our next Scribblecast. I'm Dr. Brian Lloyd. This is my Scribblecast. Thank you very much.